All right, here we go with part 2. So transcription has already happened. Transcription is the using DNA as a template, as a model to make RNA, and it makes 3 different types of RNA: messenger RNA, tRNA, which is transfer RNA, and ribosomal RNA, or rRNA. This RNA, specifically the messenger RNA, if you want to think of it as a language, it's a language where all words are exactly 3 letters long. So the dog ate the rat. Not a very exciting sentence, but it gets the point across. Those words that are all 3 letters long in the genetic code are called codons. They're a triplet of bases, 3 bases long, triplet, and they are called codons. And only in RNA are these sets of 3 bases called codons. So we make up all the sentences in this language. A sentence in this language is a protein, or also known as a string of amino acids. And you read these codons in the messenger RNA. We'll talk about tRNA and rRNA in a little bit. But we're going to be looking, first of all, at the messenger RNA and the codons in the messenger RNA. This is a chart that is going to become very, very familiar to you. You're going to be using it quite a bit. So if a codon is a set of 3 bases in messenger RNA, different charts will be set up a little bit differently, but on this one, you see here, whatever the first base of the codon, U, C, A, and G, you look at the first base of the codon here. So let's say that we're using UCG. So the first base of the codon is U, so you go right here. Then the second base of the codon, you look at where it is across the top, so C. So U and C connect right here. And then UCG, you come over here, and that's codes for the amino acid named serine. I'd like you to point out that there are 4 different codons, 4 different sets of 3 bases that all code for the same amino acid. They all code for serine. And oh, but wait, if you look down here, here is serine again with 2 different codons. So serine, you can actually get serine from 6 different combinations of codons, or 3 different combinations of 3 letters in the messenger RNA. If our codon were GAU, first base of the codon is G, second base is A, third base is U. GAU codes for aspartic acid, and each of these codons, which code for an amino acid, that amino acid has a 3-letter abbreviation to it. I think to stay consistent with the codon thing, and also because we want to abbreviate as much as possible. So the codons are always going to be read one at a time. You're not going to skip any codons, and you're not going to skip any bases. You're not going to skip any codons. You're not going to overlap anything. So when I'm reading this sentence right here, I would not read it as don't skip p or overlap any bases. That just would not make sense. I'm also not going to read this as don't kip or overla bases. I'm going to read it just as it is from left to right. Don't skip or overlap any bases. So just go left to right, don't skip any, don't overlap any, because that would be considered a mutation, and we'll talk about that later. Another thing that you need to know is that AUG is always the start codon. There might be bases over here to start the messenger RNA, but if they are not AUG, you ignore them. You just find the first time that AUG shows up, and you start there. So every sentence in this language starts with AUG. It would be like writing an entire book where every sentence starts with the word the. It'd be a boring book, repetitive, but you could do it. So there are also 3 stop codons, and they are UAA, UAG, and UGA. They do not code for any codons. This is just you know, like we talked about in transcription, it's the roadblock that causes the process to stop. That's what these do. And again, each codon corresponds to an amino acid, and as I showed you with the serine, each amino acid might have more than one codon associated with it. Now let me show you something on this chart, because most charts have this on here, and it's a nice little reminder. Hope you don't mind, I'm going to be jumping back and forth between pages a lot. If you look over here, in this particular chart, AUG is in green, and it says right down here, start codon. So it's a reminder to you to be looking for that. And then the stop codons, the 3 of them, are in red, and they say stop codon. There's not an amino acid that goes with them. So be paying attention to that. So let's give it a try here. Here is a messenger RNA. I don't know how many bases it was. I just kept putting bases in until I decided to stop. So remember, you always want to be looking for the start codon, which is AUG. The first codon, first stop code or start codon, that is AUG. So right here you have AUG. You ignore these bases over here because, well, they're not AUG. So this is going to code for methionine, which is abbreviated MET. Then the next codon is AGG. Now, I know you probably noticed that there's an AUG right here. It's not the first AUG. You always go with the first AUG as your start codon. So this is not actually going to be another methionine because we don't skip bases, we don't overlap bases. It's not going to fall together as one codon. But let's take a look at AGG. So A is the first base in the codon. G is the second base, so we're going to find where A and G connect. And then AGG codes for arginine. So this second amino acid, I'm really not good at curvy letters on the trackpad. Okay, arginine. Well, the next codon is CAU because we don't skip any bases, we don't overlap any bases. What I want you to do is to pause the video for a minute and see if you can figure out what this next amino acid would be. CAU. C-A-U. Histidine. So this next one is histidine. Wow, that was bad. And then the next one, GUU. Pause the video. All right, here's G, here's U, and here's U. So now, go through all of these. Here's ACC. ACC is threonine. So histidine, valine, threonine. The next amino acid in line is GUA. Pause the video. GUA, there's another valine. You can have the same amino acid lots of times in a protein. Then CGU is the next one. Pause the video. Okay, CGU codes for arginine. GCC is the next one, which GCC is alanine. And then here we are with the stop codon, the UGA. And if you were to go to the chart and look at this, U, G, A. It says stop codon right there. So that's how you would know where to stop. Well, not only do you need to have the messenger RNA in order for translation to take place, but you also, the messenger RNA with the codons that are read and translated. You also need tRNA and ribosomes, and I want to take just a couple minutes to introduce you to those. I know I've got an arrow out of sorts here, out of order, so just bear with me. So this is what a tRNA looks like. T stands for transfer, tRNA. I also think it kind of stands for the shape. It looks like a letter T. But this is an RNA that's able to fold up on itself. So it's a single strand. If you look at it, it's one piece that just curves around, and then there is some base pairing happening where you see these dotted lines. Because this is RNA, it still is going to be transcribed from DNA. This is the arrow that's out of order. Up here is where the amino acid attaches, which is linked to the anticodon, which is down here. Now the anticodon, because it's a codon, that means it's a sequence of 3 bases. Anti means opposite to or complementary to the codon. So in this particular anticodon, what's complementary to U? Well, that would be an A. Complementary to G is a C. Another G. Complement is a C. So if you were to go back to our chart and look at, we're not going to look at what the anticodon is. We want to look at the codon. The codon is ACC. ACC stands for, or codes for threonine. So this amino acid up here would be threonine. Then the ribosomes, remember the ribosomes are made of rRNA and proteins. The rRNA is transcribed from DNA. Ribosomes are put together, assembled in the nucleolus of the nucleus of the cell. And there are 2 parts to the ribosome, the large subunit and the small subunit. And I know this is going to be a shock, but this piece right here that's bigger, this is the large subunit. And then this piece right here that is smaller is the small subunit. I know it's a shock. All right, that's all I'm going to do for this video, and we will get to the actual steps of translation in the next one.